Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, entitled Optimize Your mRNA Process from pDNA Template to LNP Encapsulation. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Blaž Bakalar, who will discuss how the PATfix mRNA Analytical Platform enables reliable at-line insight during process development and production of mRNA therapeutics. After the presentation, we'll have a live Q&A session, so please do feel free to pose your questions to Blaž using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics covered today. Now, I'd just like to briefly introduce our presenter. Blaž Bakalar completed his BSc in biochemistry at the University of Ljubljana, Slovenia, with a focus on laboratory methods in the pharmaceutical industry. He obtained his PhD at NTU Singapore with a focus on nucleic acid biophysics. And in 2020, he joined Sartorius BIA Separations as the PATfix product manager. So without further ado, I'll hand over to Blaž now to kick off with his presentation. Over to you, Blaž.
Thanks for that very nice presentation, Charlotte. Can we just confirm that my screen is being shared?
Yep, looking great.
Awesome. So thanks for joining everybody today, and we'll be exploring how analytics can support your process development, going all the way from pDNA from E. coli to mRNA encapsulation. In the agenda, we're going to talk a little bit about process development, what's important in process development, and a little bit about nucleic acids, so biophysics of nucleic acids and similar. Then we're going to talk about pDNA purification monitoring and linearization. And then once we have our pDNA linearized, we're going to jump into in vitro transcription yield and productivity optimization, how to monitor capping efficiency, and our newest method that we're developing, dye-free method for encapsulation efficiency monitoring. So, when doing process development, there's quite a few things to consider. First of all, I would say is the critical material attributes of your inputs. So that would be the stuff going into your process. Then the critical process parameters get defined and applied so that you hit your critical quality attributes. But more than linear, this is actually a circular process where quality by design is important. Besides CPPs and CQAs, other things that are important and must be considered when developing a process with your particular circumstances are, of course, costs. Each process step offers a few solutions, so picking the solution that fits best your needs is important, as well as your existing infrastructure that presents you with opportunities, but as well as limitations. Other things to consider are, of course, recovery of each step and the process's total scale-up potential and process limitations, where specifically the way you have your facility set up will, of course, influence how you want to set up your process. So for each process development team and for each molecule, these considerations will be different. And this is why process development is usually a very lengthy thing. It takes years and years to develop a good process and transfer it to production. A lot of this is because optimization runs are done, samples are collected. Sometimes you might be limited even by how many samples you can collect at a single time for a single optimization run. Then these samples are sent for analysis, and in the meantime, you might be doing some other optimization run while you wait for the results of that analysis to come back to you so you can act on it. On the other hand, if you bring biochromatography analytics at line while you're developing your process, your optimization run delays can be severely shortened. What I mean by that is in a single day, you can do optimize a unit operation, collect samples, analyze the samples, and by the next day, you can already decide in which directions you might want to move next. And this is where, of course, PATfix can support you at Line. Now, looking at our molecules of interest, when it comes to pDNA, usually it's manufactured in E. coli. Then it's harvested, pelleted, resuspended, lysed, and with lysis, we start the purification process. Now, the interesting thing is when you lyse E. coli, of the dry material, only roughly 3% represent your pDNA, while there are many other impurities present in the lysis sample, such as proteins, RNAs, chromosomal DNA, and other nucleic acid species. Now, all of these have absorption and can interfere with your measurements, especially early in the processing, early in the process stage. While if we move forward towards RNA, it's also a very interesting molecule of nucleic acids, and they're usually quite big. So here on the right, I've highlighted it next to an IgG, and next to an IgG, even a very short RNA like EGFP is twice as big as compared to the IgG protein itself. So with such increase in size, new chromatographic techniques are, of course, recommended. And when you choose your chromatographic techniques, some of the characteristics of mRNA that you can target are, of course, the phosphate backbone, the bases themselves, the poly-A tail, the hydrogen bonding potential of the bases. And with these, you can come up with innovative ways to separate it from the other nucleic acid species. Another thing to mention is that mRNA by itself is usually not completely single-stranded in solution, but it has a secondary structure which is basically dynamic and is in some sort of an equilibrium depending on your solution conditions. And this is an important consideration when you're measuring your mRNA with UV, because UV absorbance of nucleic acids, and especially mRNA, is affected by the buffer. What I mean by that is salts and pH can influence the extinction coefficient of your mRNA, which may lead to you wrongly presuming how much mRNA you might actually have because the buffer salts have changed. Another one, of course, is that dyes have off-target binding that can skew results. So if you have a lot of impurity that has potential to bind your dye, even though it's very specific, you can still get off-target effects and wrong readouts. And both of these can contribute to erroneous readouts that can lead to wrong conclusions while you're developing your process. That's why we strongly advocate for biochromatography, which focuses on separation of your species before you quantify them. And this becomes especially important when you're trying to optimize your entire process, going from cell harvest, for instance, to linear pDNA. The earlier you can determine how much pDNA you have, the easier it will be to measure how much recovery you have with each process unit, as well as how much impurities you were able to remove, such as RNA and proteins. Similar, of course, applies with with mRNA. But with mRNA, usually it's a little bit easier to measure because it's not as dirty as pDNA coming out of E. coli. So if we look at alkaline lysis, there is a lot of potential to optimize your process at this point. The problem, of course, is there's a lot of impurities present. Here we're showing a chromatogram on the right where we've taken an early sample in the process and analyzed it to see first of all, the pDNA content, but as well the impurities that are present, such as RNA and proteins. Now, not only that, you can also quantify very early on how much open circular and supercoiled isoforms you have in this process. And with this knowledge, you can design your experiment to optimize for lysis time, how much calcium chloride to add, for instance. You can go and optimize your lysis volumes and so forth. Once you have the pDNA lysed, of course, it's time to purify it. And here we encounter our Simultus DAE prep columns, where we've put the lysis solution on the column and had three different fractions: the RNA elution and wash, the pDNA elution, and the cleaning in place fraction. All three of these we've analyzed with our PATfix pDNA platform, and the data you can get out of it is, of course, the pDNA recovery, RNA removal, and OC pDNA content, so in percentage. This allows you to have a very robust way of tracking your recoveries and process performance. Another thing, of course, to mention here is that with RNA, removing it before you go into your mRNA process is of critical importance. And finally, you can not only analyze your fractions, but you can also do kinetic work with this. For mRNA, circular pDNA is not the correct version that you want to use. You want to linearize it before you start your IVT reaction. And PATfix pDNA platform is a great tool to help you achieve just that, because you can track all three important species that are present during linearization. So your open circular and supercoiled isoforms, as well as the linear pDNA isoform. With this, you can optimize your process times, your volumes, your temperatures, as well as, of course, enzyme concentration. And enzyme concentration, of course, being one of the cost drivers. If you can get it as low as possible and still have all of your pDNA circularized, that's going to lead to cost savings and good process optimization. Once you have your pDNA linearized, you, of course, move on towards the fun part of the mRNA, which is the IVT reaction. Now, the IVT reaction can be set up in different ways. You can, of course, have a work stream where you have a two-step reaction, where in the first step you synthesize the mRNA, and then post IVT you add the cap. Or you can have it all in one-step reaction with the capping analogs. Besides the capping analog, of course, and the linear pDNA template, other things that are present in the IVT reaction would be the RNA polymerase, other nucleotides, capping reagents, salts, pyrophosphatase to remove your pyrophosphate, which is created when nucleotides get incorporated, as well as RNA inhibitors and other adducts. But out of all of these, the most important to track are your capping reagent, your nucleotides, your template, and your mRNA. And this is exactly what we enable with the mRNA platform or with our CIMac PrimaS multimodal analytics. On the left, we're able to track and separate both of the most popular capping reagents, which would be CleanCap as well as ARCA. Next to them, we can separate the nucleotides, the linear template, as well as the mRNA. And for this, we've also developed an mRNA platform based on around PATfix, where we validated the entire method, which allows you basically to take this and implement it in your site without doing method optimization for yourself, or at least starting from square one. The cool thing is it comes with SST validated standard as well as mRNA standard, so you can do calibration curves and get absolute readouts of your mRNA being synthesized. An example of capping reagent and nucleotides, which are important to track in the IVT reaction, is shown here. So when you start your IVT reaction, your conditions will be very similar to the graph on the left. As you can see, ARCA and NTPs are roughly in the same proportions. But as you progress with your IVT reaction, nucleotides get consumed, while ARCA, as a capping reagent, doesn't get consumed almost at all, just a little bit, depending on your mRNA size. But even if we can increase this ratio to 10:1, which would basically mean your IVT reaction is almost depleted of nucleotides, we can still clearly separate all of these species so that they are easy to quantify. The result of this is that you can track all of them in one chromatogram, so your capping reagent, your nucleotides, and mRNA, and do a time course to see what the reaction kinetics actually are. Time course shown here. As the IVT progresses, mRNA gets synthesized, and at a certain point, it stops. The cool thing that you can do with the platform is, in this case, you can pretty easily identify that the IVT reaction stopped because we ran out of ATP in this case. But those are basic examples. We can go further than that. You can set up a few reactions in a very small volume, 20 to 100 microliters in an IVT thermal shaker, and track these reactions at different conditions to basically extract reaction kinetics. Not only that, you can also periodically feed nucleotides, additional nucleotides, into the IVT mixture, thus keeping the IVT reaction going more further, basically allowing you to reach very high yields. Once you have these reaction kinetics very well defined, you can, for instance, then transfer them to an amber machine, in this case, where you can then perform the IVT on a larger scale and see how your model scales from small to large. Further than that, we can go towards continuous feeding and continuous production of mRNA. What we've done in this paper using PATfix mRNA platform and Amber reactor system was that we set up the initial reaction where the nucleotides got consumed, and then based on the kinetic profiles that we determined, we set up the feeding of nucleotides so that nucleotides in the reaction would stay stable around a certain point. As you can see here, they're pretty much stable until we turn the feeding off, and then they get consumed. And the mRNA pretty much comes up almost to 12 milligrams per milliliter in the final, as a final yield. The cool thing about using continuous feeding is that you utilize your capping reagent much better, you utilize your T7 polymerase much better, as well as your linear DNA template, as you can see here on this graph on the right. We've been able to reach a transcription factor of over 400, which basically allows you to utilize your pDNA template very efficiently. All of this, of course, is lowering your costs. And all of this you can read at the paper that was published just last year, at the end of the year, on the reference shown here. So once you have your mRNA synthesized, one of the common things to check is to check your capping efficiency. In this case, we've adopted a test that was published in 2017 by Chiron et al. That you can find here, where the mRNA is split into two samples, and one of them is treated enzymatically with these two enzymes, and all of the mRNA that is not capped is degraded to nucleotides, while the capped mRNA stays intact. These two samples can then be analyzed on the CIMac PrimaS column, and the peak area of mRNA comparison between digested and non-digested mRNA will let you know what your capping efficiency was. In practice, this would look something like this. In the first graph here, we have an example of the blue line, which is without the nuclease, where we see pure mRNA. And then when we add the nuclease, the red curve, as you can see, is not here. It's totally digested, and the free nucleotides come in the flow through of the method. While if we move on to the capped mRNA, even it was very well capped, as you can see, because the blue and the red curves pretty much overlap in area, which can be seen on the graph here on the right, where we can see for all three mRNAs that we analyzed, the capping efficiency was very high. So combining these findings, and with ARCA, this is especially interesting because with ARCA, as you know, you need to keep your GTP concentration low so that ARCA can get incorporated as the 5' capping agent. So we've set up a few examples here, where in the first example, we set up a standard concentration of nucleotides. And as you can see, the yield of the reaction was 3.7 ng/ml. And without the capping reagent, we didn't see any capped mRNA, of course. Then we tried going for very high mRNA expression, but as we see, the capping efficiency suffered. Conversely, if we try to go for very high capping efficiency, as you can see here, the mRNA yield suffered. But using PATfix and AMBER to continuously feed additional nucleotides, you're able to achieve both very high yields and good capping efficiency with ARCA, as is shown in the final example here on the right. So very nice. Now we have our capped mRNA, and in this process, we've gone from raw material control, where we've checked our pDNAs, we've monitored our IVT reaction, we've checked our fractions for mRNA purification to see and optimize the capture and polish steps. And you can use PATfix as an in-process analytical tool, as well as a final drug substance tool. But drug substance for mRNA is not end of the journey. You also have the final drug product, which means that you have to encapsulate your mRNA. Usually this is done in LNPs by mixing the lipid mixture with the purified mRNA, which forms lipid nanoparticles, and these particles can then be formulated and administered to patients. To monitor this encapsulation, we're developing a new method to capture this final process step where you can optimize for further gains in yields. This method does not require any dyes. It's a double column setup, and it allows you to do at-line quantification with immediate feedback. Here, I've split the method in 3 segments, so it's easier to follow. In the first segment, we load the sample, and the LNPs are bound to the first column, while the the free mRNA gets bound to the second column. Then we elute the free mRNA and measure its peak area. This would be a peak that would appear here in the method. We proceed with opening the LNPs and capturing the encapsulated mRNA, which has now been freed, on the second column, which is shown by this segment of the graph here. And then we finish by measuring the encapsulated mRNA, which would be a peak that appears here. So what kind of data can we get from this method? Here we've set up a few samples. The first sample being we injected free mRNA, so non-encapsulated mRNA on the column, on the system. As you can see, all of the mRNA eluted here in the first part of the method. And if we check the second part of the method, we see there is no mRNA present here, which of course means that there was no encapsulated mRNA present. This is something we of course expected. Conversely, if we inject only LNPs, we see that there was no free mRNA in this case, while the encapsulated mRNA can be seen as a peak appearing here after we free it from the LNPs. And finally, if we inject a sample of free mRNA together with LNPs in a 50-50 mixture, we also see corresponding peaks for both. This would, of course, allow you to track encapsulation efficiency and optimize your encapsulation conditions, as well as your lipids, flow rates, temperatures, pHs, and so forth. So putting everything together, it's very important to say that having good analytics from the very start, even if you have pDNA coming from outside, everybody's doing a linearization usually in-house. So having pDNA having trackable isoforms is important to track them, as well as separate them from other contaminants that might be present, especially RNA. With mRNA itself, it's a dynamic structure, which should always be taken into account. So even though mRNA is similar, no two mRNAs are exactly alike. And of course, the buffer conditions can have an effect. That's why we always advise, when doing analytics at least, to do separation of these species before quantifying them, because the absorbance of nucleic acids, especially mRNA, has matrix dependence. And dyes can have off-target effects. So pDNA is a critical raw material. You can use the PATfix pDNA platform to monitor and optimize your pDNA purification process, as well as track SCOC content and ratio. Not only that, you can also use it for pDNA linearization monitoring, so for kinetics, where you can also track the linear pDNA isoform. And all of this works even for very large plasmids, so over 10 kilobase pair. With mRNA, what we offer with our PATfix mRNA platform is to track IVT conditions, so all of the key components, determine and optimize synthesis kinetics, and you can get, because of that, double or even triple your of the yields that you currently might have. You can use it to track mRNA purification during your purification steps to make sure that you remove all residual nucleotides, capping reagents, potential abortive transcripts, as well as other contaminants such as your DNA template. After the mRNA is synthesized, you can use it to monitor capping analytics, as well as later on, we will also enable you to monitor mRNA encapsulation analytics. And with that, I'd like to thank everybody for attending, especially, and from our side, the PC4 team led by Andrea and the PC2 team led by Rok, which have been the major contributors to this work, as well as our managing director, Blaž, for enabling us to give you this presentation. So thank you, everybody.
Wonderful. Thank you very much, Blaž, for a great presentation. If you could just stop sharing your screen now and turn your camera on and make sure you're unmuted. We're going to start our Q&A. So I can see we've had lots of great questions coming in. I think the first question I've got here, what is the efficiency of disrupting LNP particles on the column?
Oh, that's a very good question. So from what we've seen, it's over 90%. Actually, with Triton, all of the particles get disrupted and opened. While when it comes to mRNA recovery, we are already at over 90% repeatedly.
Wonderful. And have you been able to do mass balance for this to ensure that you aren't losing anything when running through the column?
Yes, we've actually done that study. And as I mentioned, the mass balance was over 90%. So what we put on the column versus what we saw is already over 90%.
Brilliant. And can you monitor if you have multiple RNA species?
This is something that we're still implementing in our method because it's of high interest when it comes to, for instance, CRISPR-Cas mRNA therapies, where you have the guide RNA as well as the Cas9 RNA. And this is something that we're looking to do as well, to separate both, to see how much of each is encapsulated in the LNPs.
Thank you. What was the scale of the fed batch production experiment?
Oh, the one that I showed where we used AMBER and PATfix together, the volume of the bioreactor was 250 milliliters. We started at, I think, around 150 milliliters. And then as we fed additional nucleotides in, I think we reached over 200 at the end. A milliliter scale. And that would put our yield, final yield at over 2 grams of mRNA synthesized this way.
Brilliant, thank you. And does PATfix have MALs?
Yes, actually it comes with MALs, and this is also something that we're developing as an analytical tool to also help further characterize LNP particles, not just encapsulation and efficiency.
Interesting. And good question here from a viewer. Is it possible to individually measure the NTP concentration throughout the IVT reaction?
Yes, actually you can measure all of them. You can measure all of the NTPs as well as the capping reagent throughout the IVT reaction.
Thank you. And another question here: what concentration of linearized pDNA would you need to be able to achieve mRNA concentrations of 12 milligram per milliliter, as you showed on slide 18?
Oh, I don't know that one off the top of my head.
No problem. Well, viewer, we will try and get back to you by email on that one.
So if we look at it with the transcription factor at 400 and reaching 12 mg/ml, you would be able to achieve 1 to 100 easily.
Okay, brilliant. I keep the questions coming in, by the way. We've still got plenty of time to answer questions, getting lots coming in. But any that we don't get to today, we'll try and answer by email. So do keep those coming. Next question I've got here. Can you speak more to the role of UF-DF, the 3 stages in your process diagram? Are they intended to remove something specific that might cause problems in the next steps, or are they just for general sample cleanup?
So we use UF-DF for a few purposes. First of all, it can be used to change the buffer. This is important if you're doing chromatography and you want to be in binding conditions for your sample. Secondly, it's to remove impurities, of course. Like if you're purifying mRNA, which is very large, it's very easy to this way remove residual nucleotides and capping reagent, for instance.
Brilliant. Just a note to the audience, if you can't see either or both of us, not to worry, I think we're having a slight technical issue, a bandwidth issue, so not sure if you can see us on video, but as long as you can hear us loud and clear, I'm sure that's fine. Okay, so next question here, Blaž. So for the fed batch mRNA production, you improve your yield, but do you see any increase in dsRNA levels compared to non-fed batch production?
So for this one that we didn't see any increase. There was also some interesting stuff that I saw at a conference recently when we've been playing around with limiting UTP concentration to bring the potential for double-strand RNA down even further, based on some very old research.
Brilliant, thank you. Next question here. You introduced the PrimaS column for analytic use, and the separation results in your example looked very sharp, NTPs, capping, and mRNA, etc. Is this multimodal chromatography resin also usable for large-scale purification of mRNAs?
Oh, that's a very good question. Yes, we actually offer PrimaS for mRNA purification on a large scale as well. And we have quite a few clients using it already in that respect. It's especially useful, for instance, if you don't have a poly-A tail in your RNA.
Wonderful. Yes, that was a great question. Another question here. What is the frequency of data collection for continuous IVT reaction monitoring?
So it's limited by the method itself, and the method takes 8 minutes. So the lowest you can bring it to is 8 minutes. I think we usually do 15-minute time points, though. Usually that suffices to get the relevant information.
Brilliant, thank you. How do you separate dsRNA and ssRNA?
So for our purification process, we usually offer 2 solutions here. One is based around SDVB chemistry, the other one around C4HLD chemistry. For analytics, we actually, as part of our PATfix mRNA analytical platform, we offer the SDVB method as well for detecting double-strand RNA. I haven't gone into it today because double-strand is a little bit finicky, let's say. And there's still no real consensus of what kind of double-strand RNA are we talking about.
Yeah, that makes sense. And what type of analytical method is used for analysis of mRNA or pDNA?
So for pDNA, we offer an analytical platform in 2 scales. We use our CIMac pDNA column, around which we've developed 2 methods. One is for, we call them small plasmids, which are less than 8 kilobase pair in size. The other one is for larger plasmids, and you can do quantification on those as well. So we've quantified stuff even bigger than, I think, 19 kilobase pair. So very large plasmids. And for mRNA analytics that I've been showing today, The primary workhorse, so the consumable, the column, it was the CIMac PrimaS analytical column, around which we've developed a method and the platform as well.
Perfect, thank you. Lots of great questions coming in. So next one here, I've got, how do you detect the difference between, I thought it was the same question again, but no, slightly different one. How do you detect the difference between double-stranded RNA, hairpin, and DNA? Really
Very difficult.
With great difficulty.
With great difficulty. So hairpins are actually something that is naturally present in all RNA molecules, for that matter. It's something that the RNA structure will assume, especially if the conditions, so the buffer conditions, are right, usually with a higher salt and around neutral pH. I would say that's not something we looked into, but I'd be happy to connect after the webinar and discuss this further if it's of interest.
Absolutely, yes. We can follow up by email later on. Next question got here. Why are there two peaks for the mRNA in the chromatography? Does it correspond to secondary structure? And how would you check for abortive transcripts?
Yes, that's actually a very good observation. So with our analytical method, we actually see two peaks for mRNA. What they correspond to is unstructured mRNA and structured mRNA. What we did is we collected these two peaks separately and analyzed them on capillary electrophoresis as well as other analytical methods. And what we saw is that no matter what we did, they always assumed, let's call it, the same equilibrium, the same shape. That's why I was mentioning that the RNA structure itself is in this dynamic equilibrium between structured this way, structured that way, unstructured, and so forth.
Brilliant, thank you. And next question I've got here, how can you disrupt the LNPs over the column? Is it on column or outside?
Oh, it's actually on the column itself. That's why when I saw this method being developed, I was kind of, I was kind of shocked by it, like very cool. So the LNP particles get bound on the first column. I cannot disclose what chemistry it is though. And as they're bound on the column, when we introduce Triton, it will naturally disrupt the lipids that encapsulate the mRNA, which will allow the mRNA to be freed and then detected separately in the last part of the method.
Brilliant. And could you provide information on what type of bioreactor was used for the fed batch?
So for fed batch and continuous feeding, what we use was an Amber system. I think it's a very common thing, and actually quite a lot of clients already utilize Amber for these purposes, especially for IVT.
And I think we might have touched on this already, but for the fed batch mRNA production, you improved your yield, but do you see any difference in innate immune activation compared to non-fed batch production?
Okay, that's a good question. So from the stuff that we can show here that's done on our own material, we haven't done any cell studies yet on it to basically confirm or disprove on what sort of effect it might have on the innate immune response of cells.
Brilliant, thank you. And during the fed batch or continuous IVT for mRNA, what's the maximum concentration achieved for the mRNA, would you say?
Yes, the maximum we've been able to achieve, I think, was around 12 mg/ml. But we need to have that with a caveat. Because we were continuously feeding additional volume of nucleic NTPs and salts into the reaction chamber, we were actually constantly increasing the reaction volume as well. So even though the concentration-wise, we've been able to reach 12 mg/ml, in pure sense of how much we were able to produce, it was very high, as you would be able to achieve in such a starting volume.
Brilliant. Lots of great questions coming in. And here's the next one. Could you assess the exact purity or integrity of the mRNA produced?
Exact purity and integrity. Yes, we actually offer 3 different analytical methods within our PATfix package. And the cool thing about them is they are orthogonal and they probe different characteristics of the sample. So besides PrimaS method, which can be used to actually quantify potentially residual nucleotides as well as residual template, DNA template, we also offer oligo-DT analytics in the package, which is a much simpler method, but gives you orthogonal information because the only things that bind to the column are mRNA molecules that are fully synthesized and intact because you require a poly-A tail to bind to the column. And lastly, we also offer a SDVB method, so reverse phase method, which we utilize to separate by size. So with the combination of these 3 methods, you can have a very good understanding of your purity as well as integrity. Sorry, are we still on?
Oh, apologies. Question here from a research scientist. He says, is it necessary to use a second C4 HLD column if we're not linearizing DNA?
That's a question of preference. So the C4 column that we use in our production process, or recommended production process, I would say, because for everybody it can be a little bit different, is to enrich the supercoiled isoform, for instance, from the open circular isoform. So it might not be strictly necessary, but it is important that you're very sure that you've removed residual RNA, for instance, before utilizing it for a reaction like IVT.
And is it possible to isolate or enrich for full or non-empty in INPS?
Oh, okay. So this isn't
LNP,
something.
sorry. I was misreading.
LNPs, full or empty LNPs.
Yeah.
This is actually something that we're kind of continuously looking into. And we just started with this question. So there's not really a lot of information that I can speak to on this webinar.
Okay, no worries. And another question here. Is there any impurity, say dsmRNA, observed during the fed batch mRNA? We covered that a bit already, but if you don't mind.
Yeah, I think. So we have an analytical method that is able to detect this double-stranded RNA, which we've confirmed with the J2 antibody as well, which is the standard in the industry right now. But I think I already answered, sorry, that the continuous fed batch, continuous feeding didn't increase the double-stranded content.
Mm. Wonderful. And can we also have information on the, I think it's again a similar question, quality attributes of the mRNA like dsRNA, poly-A tail length, etc.
Sorry, can you repeat the question?
I think it's another similar question asking about, they're asking, could could we have more information on the quality attributes of mRNA, like dsRNA and poly-A tail length?
Okay, so for DS, I think I already answered quite a few times.
Yeah.
But for poly-A, we're still considering if we should go in this direction or not. But if you're interested to strike a collaboration around it, we'd be happy to explore that option.
Brilliant. And another question here, is there any analytical method for measuring full-length transcript? Again, I think we touched on this already, but...
Yes, the full-length transcript will, of course, have a poly-A tail, usually, if the poly-A tail is included in the template as well. It's very easy to do it with the oligo-DT analytical method.
Absolutely. And methodological question here. What percentage of Triton X do you use to disrupt the encapsulated mRNA?
Yeah, I won't answer that one. Next question, please.
Okay, no problem. Well, I think we've got time maybe for one more question. Is 12 milligram per milliliter a specific value, or is it possible to go beyond that value? Is there a limitation to reach only, can you only reach up to 12 milligram per milliliter?
Yeah, that's a very good question. At a certain point, you have to ask yourself, does it make sense to go higher than that? Already at 12 mg/ml, the solution itself is quite viscous. So going beyond that, how it would influence your process going downstream, I'm not sure. I'm not sure. Maybe, but probably, yeah, maybe.
To be determined.
To be
Brilliant.
determined, yes.
Brilliant. Well, thank you very much, Blaž, for answering those questions. That's all we've got time for today. So any questions that we didn't get to, we'll try to reply by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left is to thank Blaž once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you.